We have a fantastic speaker now coming up to talk about innovations in synthetic biology. He needs very little introduction for many of you, but because, there probably are some people that need to know, here is his full title, Termier Professor of Medical Engineering and Science at MIT. But he's also a co-founder of Antibiotics, Synlogic, and through his lab of multiple other startups, and the lab is called the Collins Lab, and Sample 6 is another of those companies. So without any further ado, Professor Jim Collins.
Thanks, Tron. Well, I want to thank Tron for the kind intro and more so the invitation to speak to you today. I'm really delighted to have the opportunity to share with you a new area of synthetic biology. So you've already heard this morning, this fascinating, relatively new field has been bringing together engineers and biologists to basically create synthetic gene circuits and other molecular components and use these primarily to rewire living organisms, endowing them with novel functions for a range of applications. And we've heard several of these, and really it goes back to the idea that could you now create these circuits just as an electrical engineer would? And just as the engineer might model it, could we also use mathematical modeling? We heard about Asimov, a recent spin-off from MIT that's going to dominate that space. And now just as the engineer might go get resistors, capacitors, inductors, could we now go get genes, promoters, other bits of DNA, RNA, and proteins, and instead of soldering them onto breadboards, could you use the tools of recombinant DNA in a cut and paste fashion to create plasmids and reinsert these now into cells, giving them these novel sense, decision-making, and respond capabilities. We've already heard about these, but these kind of programmable cells are impacting multiple industries. And as Tron mentioned, I'm a co-founder of Antibiotics, which we heard from Jeff earlier, where he's taking engineered phage and using these to get after infections. I'm also involved with Sample 6, which is using engineered phage for food safety diagnostics. And Synlogic, which was on the schedule, but unfortunately wasn't able to come with J.C. Gutierrez-Ramos, is using engineered bacteria as living therapeutics, going after rare genetic metabolic disorders, inflammatory bowel disease, and increasing our immuno-oncology. What I want to do is tell you about a new area which is maybe of interest to many in the audience from a material standpoint, and that is, could you, instead of using living cells, nonetheless harness the power of synthetic biology in new ways where you don't have a living cell present? So again, you can think of many applications, whether it's in your home, other consumer settings, or even in a hospital, where you don't want to have a living cell. We got excited about this when Keith Pardee in my lab began thinking about and looking at cell-free extracts. So what are these? These have been dominant in molecular biology for several decades as a discovery engine. What you can do is you can open up a living cell and take its machinery out. So that machinery would be several dozen enzymes, nucleic acids like DNA, RNA, and molecular machines like ribosomes. And typically what the molecular biology labs will then do is you'll play with this inside a petri dish or test tube. Keith, for reasons I still don't understand, actually said, well, what happens if I take that cell-free extract, spot it on paper, and freeze dry it? Can I rehydrate it sometime later and still have activity? And what he found was fascinating. He found that he could take synthetic circuits, place them in with cell-free extract, spot it on paper, freeze dry it, sometime later rehydrate it, and what he had spotted on paper would function as if it's inside a living cell. Okay, so for those of you who play with cell-free extracts, maybe we're trained on it, what you'll typically do is take them fresh from frozen stock. So you'll have your large -80 freeze, you'll take it out, you'll thaw it, you'll play with it. Keith wanted to see, did the freeze-dry process impact the activity of that in any way? And shown on the left is there was no impact on the activity. He didn't lose any activity for any of the components of interest to synthetic biology. He did an incredibly informal, non-scientific control scheme where he basically took a number of these paper-based systems and just shoved it in the drawer in his lab bench at the Wyss Institute, took it out about once a month, and we're now well past a year, and some of the early data shown here, where little, if any, loss of activity in the freeze-dried scheme when stored at room temperature, now well past a year. Hey, we're synthetic biologists. Could you do more complicated schemes? So with Alex Green, we had developed what are called toehold senses. These are RNA switches that can be engineered to sense any RNA sequence in either a clinical sample or other samples. And in many cases, you could envision not wanting to have to use fluorescent proteins, which is what Keith had earlier done. So could you instead have a colorimetric, easy-to-read system just by eye? And so Keith had the insight of replacing fluorescent proteins underneath his very complicated RNA switches with LAC-Z. LAC-Z is an enzyme when expressed. If there's another enzyme present in the substrate, will actually cleave that enzyme, and the substrate will change from yellow to purple. And what we're showing here are easy early results where Keith and Alex were able to show that on these simple paper-based schemes with RNA sensors, you could actually get a colorimetric, easy-to-read output within about 20 to 25 minutes. So it'd be kind of similar to a paper-based pregnancy test, but now you could broaden what you might actually sense on that. This work was done at the Wyss Institute in collaboration with MIT, and the Wyss is very much focused on translational efforts. And what we were able to do there was actually produce a rapid and inexpensive means, just using computer printers, of printing out basically paper-based arrays that could then be put into a high-throughput scheme to spot cell-free extracts onto them. We also made a very simple electronic reader using the LED system for about $30 or so, where you could take arrays of these, place it in, it just shines LED through, can quantify the colorimetric change, and send a signal to your smartphone or PC for more complicated readouts. So where do we go with this? Well, much of my lab, as you heard from Jeff earlier this morning, is focused on antibiotics. And one of the big challenges on antibiotics is, can you diagnose antibiotic resistance so that you can give your patient the right medicine as quickly as possible? What Keith was able to do was take his RNA sensors and very quickly develop a number of different sensors to detect mRNAs associated with different antibiotic resistance, and is able to show here that he can actually give you a readout in about 20 to 25 minutes on a patient sample. Do you actually have that resistance marker present? We were writing this up for publication now about 3 years ago, had interest from an editor at Cell, and Keith and Alex came running because, Jim, we don't want to submit the paper right now. They said, we want to take on this challenge. And so this challenge was the Ebola crisis. So this is August, September 2014, at the height of the crisis. And these guys says, we think we can develop inexpensive, rapid diagnostics for the crisis. So what did they do? Well, there are 5 nuclear protein mRNAs associated with the Ebola virus. They took one of them, used bioinformatic analyses, and actually identified 12 different regions on it that could serve as triggers for their RNA sensors. They did 12 for the Sudan strain, 12 for the Zaire strain. In 12 hours, they designed, built, tested, and validated these 24 different sensors using only $20 worth of DNA. So you think of the thousands of dollars it takes to create antibody tests. You think of the many weeks, if not months, to create antibody tests. These guys did it in half a day, and were able to get brilliant readout. In about 20, 25 minutes, they could actually discriminate between the Sudan and the Zaire strains, which was really a stunning demo. So what do we have on these early efforts? Well, what we've really done is remove the cold chain. So those of you who play with living organisms, you play with biological material, you know a big challenge is you got to refrigerate stuff. So if you want to ship it around the world, you've got to take careful care to package it. Once you get it, you got to store it in special ways. What we showed is that you can now actually use synthetic biology out in the field, in your home, in your lab, in a much easier fashion, without the need for refrigeration. You can spot these on paper or other substrates, which I'll come to in a few minutes, freeze-dried, stick it in your pocket, get on a plane, and take it out anywhere around the world. You can stick it in a regular envelope and send it around, and then store it at room temperature in developing countries or in developed countries. And this is really cheap. If we use the high-end cell-free extracts you might get from NEB, it's still under $1 a sensor. And we're now working here at MIT to do homebrew cell-free extracts. We're now down to 2 cents per sensor. You get colorimetric readouts. I shared one, LAC-Z. We now have multiple other encodable dyes, so you can have multiplex schemes. And again, kind of get after the pregnancy test type scheme, but now for a broad set of diagnostics. And we've coupled it to inexpensive optical readers if you want more quantitative. So where do we go? I want to give one anecdote and then talk about things we're going to right now, as well as materials. And the anecdote is on Zika. So about 1.5 years ago, members of the MIT community, including myself, received an email in late January from MIT leadership asking who was working on Zika. The crisis had just appeared in the public forum. I had just learned of it the week before. And they said, who's working on it? We need to have a concerted effort. We were not. We're not a virus lab. We're a bacterial antibiotics lab. But I called Keith and Alex, who had since started their faculty positions in other institutions, says, what do you think? Should we go after this? They said yes. I turned to a number of postdocs and grad students in our labs. What do you think? They said yes. And amazing, this team came together over about 5-6 weeks, dropped what they were doing, and actually developed an inexpensive rapid platform for Zika. We developed about 4 dozen different sensors for Zika. We teamed up with Lee Gehrke, part of HST, and David O'Connor at Wisconsin. We're shown here for pennies, and in about 90 minutes to 2 hours due to need for some amplification, we could actually detect Zika at the low femtomolar, so clinically relevant samples, and we could distinguish Zika from dengue. The bottom is actually results from monkeys. So this was in the spring of 2016, where you couldn't find human samples here in the US for testing, but David was able to infect monkeys, and this is in about 2 femtomolar scheme. We could give you readouts in minutes after you have appropriate amplification of whether the monkey actually was infected or not. So we went further, is that we actually introduced a CRISPR-based scheme. So in this case, with Guillaume Cotterell, who was a postdoc in our lab, we actually found that your first step, you could use our RNA Senses to tell, do you have Zika, or do you have dengue, or other related fever-based viruses? But we were able to use CRISPR and actually show that you could use CRISPR now to discriminate at a SNP level, so single base pair difference. Do you have the Asian strain, do you have the American strain, do you have the African strain? And further, we showed you could freeze-dry CRISPR components, so the guide RNA and the nuclease, and actually have it still function. And amazingly, for reasons we don't understand, we found that the freeze-drying in many cases increased the nuclease activity, so it made CRISPR crispier and stronger. Where we've gone with this, some of you have been tracking the story, is that we teamed up also with Feng Zhang of CRISPR fame here at MIT, and we've now developed a new platform called Sherlock that uses CRISPR-Cas13a that goes specifically after RNA with collateral activity, and we can get in the attomolar range now. And that is, we can go single molecule at a microliter and get at SNP level differences. So where are we going with this? Well, we're really excited about diagnostics. We're looking at antibiotic acceptability. We're looking at HPV, hepatitis C. We're doing a tuberculosis sensor. We're moving into cancer diagnostics. We're looking at microbiome. On the education side, I'm really excited to hear Natalie's marvelous work. We're looking to see, can we use this as a basis to create a chemistry kit equivalent for synthetic biology? So people of my generation remember the chemistry kits that were fantastic. You could blow stuff up. You could combustibles. You could kill things. Now you can't do it. They neutered our chemistry kits. But our hope is, can we create a chemistry kit for synthetic biology so we can help folks learn synthetic biology at home, maybe coupled with the BioBuilder, as well as in middle schools, high schools, and colleges, and do it in an abiotic, so moving living cells. And then finally, I want to share this really exciting space, is we also recognize you could use this for portable molecular manufacturing. So what's that? Well, I made some allusions, and that is that this platform is not limited to paper. It actually works on any porous medium. So it works on plastic, quartz, glass, cloth. I'll come back, make a comment on that in a second. And it, in fact, doesn't need a medium. You can freeze dry these cell-free extracts. You can freeze dry your synthetic biology components as pellets. Carry them in now small plastic vials and simply add them to water to make biomolecules of interest. So what we've shown, we published about a year ago, is you can use this to do on-demand biomolecule manufacturing. So we showed you could use this to make antibodies, nanobodies, antimicrobial peptides, vaccines, small molecules through multi-step metabolic pathways, phage and viral vectors now. And so the notion is you could use this for a military personnel or a hiker or an athlete or a health worker in the developing world or an astronaut heading to Mars or stuck on Mars, where give him or her just a small packet where you have hundreds of different vials that could be added to water to make what they need on the spot as scheme. Two related schemes, and I'll end with this, is we also are looking to see, incorporating this technology as well as the diagnostics, to create a new class of smart bandages. So can you use this, for example, now as part of wound dressings to produce small molecules that will help heal the wound, as well as report on the state of the wound? So do you have an infection? Is that infection resistant? And then finally, an area I'm very excited about is we're using this as a basis to get after wearable synthetic biology. So can you embed biological components that are appropriately freeze-dried in clothing and potentially do two things: report on the physiological state of the person. Are they working out too hard? Are they stressed out in the field? And as well as, have they been exposed to something? So have they been exposed to a nasty pathogen walking on the wards doing their rounds at the hospital? Have they been exposed to fentanyl? Have they been exposed to something other nasty? And it's an area that we hope will actually lead to a new set of technologies that will excite people. And my clock just hit 0, and I just want to recognize the outstanding groups of young synthetic biologists whose exciting stuff I got to share with you. Thank you. Do you want me to throw that box around?
We could take a question.
Yeah.
Why don't you throw it around?
Okay. Who has a question?
But that's not what we do.
Do you throw it?
You just throw it.
Oh, sorry. Sorry.
He's not trained yet.
Yeah, no, I'm not trained, no.
When you talk about field deployment, is there concerns in transcription and translation being exposed to UV and methylation of the DNA in the circuitry?
It's an interesting question. We haven't had concerns or seen too much on that. We've left it out as of yet at the levels that you might see. And so, UV will obviously damage DNA and lead to schemes. What happens, and where we're not so concerned, is that we have a lot of material that we put in. So you're not going to get a universal set of mutations. You'll probably get some small set that will screw you up a little bit, but you'll still be overwhelmed by the majority of stuff that shows up. We are worried about humidity.
Yeah.
So you can envision if you don't have proper packaging, your system's already going off when you walk out of the airport in Florida. If you're going to try to help folks down there and make measurements, is there cholera in any? So there you'd have to have appropriate packaging to deal with the moisture.
We'll take one more question. This is unusual, but I see the interest level here is very high. We'll take a super quick question.
Can you speak a little more about tuberculosis and how this technology relates to something like PCR-based gene expert, which is now?
Yeah, so if you're doing a PCR-based system, we're not going to beat PCR on sensitivity, but we're going to beat PCR on speed, cost, and ease of use. So I think what we have is kind of a front-end scheme. So PCR is a scheme where you can do basically sequencing on the spot to see do you have it. And I think this is where we can make a difference, particularly in the developing world, where you can take sputum and can you actually get after two questions. One is, are they infected or not? And two, do they have a susceptible strain, and to which regimen should you use? And we're working with Deb Hung at the Broad now to see, can we get after it in kind of a multiplex fashion that one could get specifically at that latter question on susceptibility.
I'm going to make one promise to the audience. You will get involved very quickly in the panel discussion. We absolutely need to move on. This is kind of like we are down to the minute and seconds here, so I apologize for that. Thank you so much, Jim.
Yeah,
and he
sure.
is part of the panel.